Hi everyone, and thank you to the Immunometabolism Forum and Dr. Finlay for giving me the opportunity to showcase my work. My name is Fagan and I am a third-year PhD student at RCSI. I work in the research groups of Dr. Annie Curtis and Dr. Oran. The title of my presentation is Keeping Time, Our Knees and Hips Need a Clock That Ticks. So my work is based on cartilage tissue and understanding the processes by which cartilage senses and takes up nutrients to maintain healthy functions, and also the processes by which cartilage breaks down or degenerates. Cartilage is a wonderfully slippery substance that lines the end of our bones and allows frictionless movement between them, as shown in the image of a healthy joint on the left here. If cartilage wasn't there, our bones would grind painfully off of each other when we moved around. When cartilage breaks down, becomes worn away or damaged, as shown in the image of a joint on the right, this results in a condition called osteoarthritis, which leads to joint pain. An important aspect of my project involves our body clock. Due to the rotation of our planet each day, one thing nearly every living thing has in common is an internal biological clock, to ensure that the activities of our cells and organs follow a 24-hour profile. Our master clock is located in our brain and connects us to our external environment by sensing light. The light-dark cycle we experience each day is the main synchronizer of our master clock and ensures alignment of our cells and organs with other cycles we experience each day, such as rest and activity cycles and feeding and fasting cycles. Chondrocytes are the only constituent cell type in cartilage. We have shown that a molecular clock exists in chondrocytes and cartilage tissue by following the levels of a specific clock gene over the course of 1 week. The levels of this gene rise and fall in a specific rhythm every 24 hours. Our internal molecular clocks ensure that our behaviours are correctly aligned with a 24-hour day. We can thank our molecular clock for the fact that most of us sleep and do not eat during the dark. Chondrocytes make cartilage tissue, which is made of protein, by consuming amino acids via Slc receptors, which are expressed on their surface. Amino acids are the building blocks of all proteins. Chondrocytes consume nutrients in order to enable repair of cartilage tissue the same way we eat to fuel movement. Of particular interest to us, the Slc receptors Slc7A2 and Slc7A7, among others, are expressed in chondrocytes. Slc7A2 and 7A7 transport arginine, ornithine, and lysine. Most interestingly, Slc7A2 appears to be cycling with respect to time of day in chondrocytes. When these particular Slc receptors are knocked down in chondrocytes using siRNA, a decrease is observed in catabolic gene expression. These are genes which degrade cartilage, for example MMP13. We believe that the molecular clock which exists in cartilage allows chondrocytes to eat at the correct time of day. Our hypothesis is that circadian expression of nutrient receptors may allow cartilage to take up nutrients at one time of day and release at another, in order to enable joints to anticipate and withstand daily movement and rest cycles. We believe that this circadian expression of nutrient receptors may result in delivery of new protein to the cartilage at a specific time of day to enable rejuvenation of the tissue. Just like with our whole bodies, having these things occur at the correct time of day keeps our cartilage and joints healthy. However, we have shown that when osteoarthritis develops, it essentially stops the clock in chondrocytes. We believe that without a functioning clock, our cartilage cells start to perform certain functions at really erratic times of day. We believe that this problem of clockless chondrocytes causes the disruption of the ability to feed and rejuvenate at the correct times of day, and this in turn is what may form the basis for development of osteoarthritis, cartilage degradation, and the joints becoming painful. The aim of our research is to understand why the clock stops working in osteoarthritic joints, and whether we can develop new ways to cure or fix the clock in osteoarthritis before it is too late. Thanks to the organisers for the opportunity to showcase my research here on the IMF YouTube channel. Thanks also to my supervisors, all members of the Curtis Clock Lab and Kennedy Biomechanics Lab. Thanks finally to the Irish Research Council for my scholarship funding, and thanks to you for watching.